now we're not copyright infringing.
Oh, that's right. And so now the video is actually going to start, right?
So now the video starts.
YouTube doesn't let us post things if we've got copyright infringement going on or playing things like that. So anyway, there you go. Welcome. Hello, everyone. I see lots and lots of things that you are thinking about: school, managing time, all of these things. I also see some stuff that I think is probably related specifically to this course. Like there's lots of quizzes and lots of assignments and lots of homework, and I feel you, there's a lot. And we did that on purpose. Let me tell you why. Because you can have like, there are a couple of strategies, right? You can have like lots of little things throughout the semester, or you can have like big single things, like one midterm and one exam. Yeah, that's a no-no for all sorts of reasons. And so what we have are as sort of what they're called low stakes, lower risk assessments to help keep everyone on track and to keep you participating throughout the semester. Now, it means that you can't just, do nothing until the midterm and then cram and then, to get through this course. And that's intentional. It may not be your preference, but the idea is that you get feedback on all of these things about how you're doing, and you can experiment with different ways of studying and approaching the material and that kind of deal. So it is entirely intentional. There's a lot, but the value of each of them is specifically meant to be lower than in other experiences in order to keep that balance. And so I hope that that makes sense to you.
I've been doodled.
You've been doodled. Oh, people are doodling on you. Look, that's amazing. Okay.
So what are you thinking?
What are we thinking?
What are you thinking?
What are you thinking? We're thinking.
I'm fine. What are you thinking?
Oh boy. Okay.
Thank you, Rosetta Stone.
Yeah. Let's just see if we can see a few others here. Balancing with your other courses. Meh. Yeah, we're feeling meh, I think is the way that. Yeah. Awful with time management. Yes. And can I just say that it's grant writing season for us. So basically, we're writing these big proposals and budgets and everything, and there is not a single one of us that is going to hand it in more than 10 minutes before the deadline, right? Time management, they beat you with it and having to do it on time and all of this stuff. But reality is, and if you ask any person that's in their job right now how close to a deadline they work, it's right up to the last minute. So I would prefer that you think about how you can sort of manage the anxiety that you get from working to a deadline, rather than trying to, beat yourself into working on the weekends so that you don't enjoy your life as well. So kind of try to flip it around a little bit. Overwhelmed, I gotcha.
Yep, everybody's stretched thin.
Look at that.
So keep keep giving kindness, expect to receive it.
Yeah.
And if you don't, say, hey,
yeah.
Where's the kindness?
Yeah, you're worried that you're going to miss something. Oh.
Oh,
yes. Oh, you know what though? We should not just have them like draw on you. Here, you can draw on me too. Okay. Yeah.
Need to work on those antennae.
Yeah, you totally need to get.
Thank you for charitably inferring what color of hair I used to have when there was hair.
Yes, and it's appropriate. Okay. Amazing. So we'll keep on checking with you. If you need to or want to discuss things, hang out after class. We can give you all sorts of, we have opinions. We can share our opinions and maybe they'll be helpful, or we can find resources. So, oh yeah, you have lots of low risk assessments in all your classes. Yeah, because it's the right thing to do, and I know it's a lot.
Yeah, I see somebody. Yeah. It's C. Witten, stressed because I haven't received textbooks. This is coming up in the chat.
You mean no textbook in this class, so that's stressing you out?
I think it was
or they haven't arrived yet.
Other courses where texts haven't arrived yet.
Yeah. Okay, you haven't received your textbooks.
Whoa.
Yeah. Whoa, that's a lot. Okay. Stop annotating, please.
Whoa.
And we are going to clear the screen. There we go. Okay, thank you very much for sharing. I hope that you were able to, if you didn't type in, but you were able to see some of the same types of worries that are going on in this giant Zoom room which spans the entire globe as well. Okay. So we want to talk about all sorts of things, kind of tying in some of the big concepts, some of the definitions. There are slides with definitions in this class.
Oh, like get your pencil ready.
Yeah, don't worry, we've written them out for you, so you don't have to. But we're gonna play with them, we're gonna use them, and so we're gonna all kind of get on the same page about some of these things. But first,
it's a week.
And it's a special week.
It's a special week. It's National Forest Week in Canada. All across the country. All the forests are out and all the trees are together and they're all swaying in the breeze and they're thinking, this is the week. I hope I don't get turned into paper.
That's terrible.
This is a view of a forest that you're going to learn about and spend some time in digitally. This is on the campus. This is part of the protected natural areas on campus. This is the Dairy Bush. And this particular photo from earlier this summer is part of a contest right now in Canadian Science Publishing called Visualizing Science. So that book face link, if you click on it and like it, you can support a forest of yours that you're going to learn about later on this term in that contest.
Yes, and these links, of course, are going to be available on the PDFs that we post if you want to go and check it out. It's pretty cool. Okay, today, review stuff, learn new stuff, practice using stuff is basically what we're going to do. So hold on.
So get your stuff.
Get your stuff. Yeah, we're going to walk you through a series of exercises. Actually, what happened here? No, we're not going to do that number 3. We're going to do this number 3. And apply them. For some reason, it kept the old version as well. Okay.
Of course it did.
Yeah. We'll make sure that the right list gets posted.
I'm going to say for the reason is 2020.
2020. Yeah. Okay, so reminder, we asked you to look into the Veliger larval cycle, and it is really different than the very cool Unionid life cycle. So this one here is perhaps maybe a bit more familiar or a bit less mind-blowing, but it has incredibly important implications, and that's what we're going to talk about today. Some of you have done your homework, some of you have been overwhelmed, so what we want to do is we want to get everybody on the same page. If you're cool with it, we have a bit of an activity to help you help each other get caught up or to share some of your ideas, okay? We want you to think about the Veliger larvae, we want you to think about the Glochidia larvae and the difference between them. And we're going to make breakout rooms and have you talk a little bit, okay? So what we'd like you to do is, if you are in breakout room number 1 to 15, we want you to talk about the advantages of being a Veliger and the advantages of being a Glochidia larval type strategy.
We've made glass half full and glass half empty.
Yes.
So you're 1 to 15. The glass is half full.
Yay, advantages. 16 to 30, both of them suck. Disadvantages. So what are the disadvantages from an evolutionary perspective or like any perspective? And then pick 1 or 2 people from the room to get onto this Google Doc. We have it posted in the chat. We can only have, apparently Google has a thing, you can only have 100 people on at a time.
I think it's a COVID thing.
It's 2020.
Yeah.
So pick a few people from the breakout room to go in and start recording the advantages and disadvantages. Are you ready? I hope so. Thumbs up? Okay. I'm going to create these breakout rooms. Blah, blah, blah. And where are they? Oh man, you should see the screen right now.
You can interpret it maybe from our faces.
Wait. Let's try this. Oh, I see you all, which is lovely now that I've stopped sharing. But what I don't see, to be perfectly honest, are these breakout rooms. That's ridiculous. Okay. No, they're really like not here anymore. Wow.
I'm just imagining how we would feel if we were on campus and there was a building that had rooms that we walked by and it's like, where's the room?
Okay, you know what? This sucks. I'm really sorry. And I'm going to send some angry emails in a little while. But what we're going to do instead is if you have a last name that starts with A, D, G, M, P, or R, go onto the Google Doc and start filling out. You have a few minutes, and then we'll switch it up with other people so that we can start filling it out together and sharing some of these things.
D,
G, P, or M, P, and R. Yeah. Go onto the Google Doc, start figuring it out. We'll put it up here so that everybody can see it. Those people who are not on the Google Doc. Here we go. Here's the Google Doc. Oh, Zoom. Oh, Zoom.
we love you, Zoom. Oh, yes,
No,
we
we
do.
don't.
We love you, Zoom. This is not true. You have screwed us this morning.
Yeah.
Ooh.
There we go.
Oh, Zoom, we don't love you.
Yeah, what happened to breakout rooms?
They're gone.
They're gone.
Gone, the big gun so long, the big gun so long. Gone, the big gun so long, the big gun so long.
I'm gonna go over here. I don't want to change anything.
I see some stuff happening in the room. So the room is at least open.
The room is open. There are people there. Hello, people! So here, take a few minutes. Oh, you know what? I don't seem to have the same link as you do. Ah, there we go. There we go. Okay, cool.
Please help me remember
Yeah.
that I would like to share what those words actually, what they're derived from, because I think that might help.
Oh, okay. Veliger and Glochidia.
Yeah.
Wonderful. Yes, I love that. Okay, cool. Okay, now, those of you who have last names that, I need to see the list so that I can not repeat any letters. There we go. Last names. So all of you who are on, please get off the Google Doc and then we'll add some more people. So Smith is going to put out some last names to give you an opportunity to come on and join us. Just a reminder, the link is in the chat. And come on ahead and add to this list. Glochidia pros. Yes.
There once was a Glochidia from Norwich.
I can't add to this in any way.
I think it's what you're experiencing is that when the list has more than 100 people in it.
I get locked out,
No, you can see it,
but I can't
you can't
join
edit it.
I understand. We're learning all of these restrictions.
We are seeing what you see.
Cool. Welcome again to a new batch of students. Here, go ahead and add what you can. There we go. That's delightful. Well done. Okay. Cool, cool. Now, for those of you who are on, if you wouldn't mind, if you're done, take a step back so that we can allow anybody else who would like to join onto the Google Doc to add anything. Please go ahead and do that. Mm-hmm. And then I'll refresh. That's not refreshing. That's going back in history. There we go. Okay. Are we getting more and more? Yes, we are. Super. Okay, good. I'll refresh one more time just to make sure. Okay, cool. So, very risky. I like it. Okay, yeah, if you want to annotate, please go ahead. And if there are any ones that you would like me to, or Smith, to talk about specifically, please feel free to put a stamp on it as we go through some of these different things. So looking at the pros of the Glochidia approach to life. Yes, it definitely does, incur some safety in the development, enables them to compete with other mussels. I'm not sure what you were meaning specifically by that, but certainly because of the fact that they are safer, then it does provide them a certain protection, which gives them a competitive edge. Host fish adapt to new environments, hence mussels do too. That's a very interesting thing related to what's that?
We don't see our video on the screen.
Oh, okay. Related to co-evolution. That's okay because they can see us. Yeah. The lures make them more attractive to fish. Yes. Can use complex lures. Yes. Protected by host fish when developing. Yes. Protection. Absolutely. That's definitely the major pro to that lifestyle. Con, they need a host. Yes, so they are reliant upon a totally different organism for their survival. Needs fish to fully develop, don't produce a lot of offspring. That's interesting. Decreased fish population means less options. Yes. Yeah.
A specific expression of the dependency.
They cannot crawl or swim because they rely on, you know what? This is overlapping. More sensitive to changes in environment because they rely on another species' survival. So yes, in kind of like a 2 degree of separation way. If that host species is sensitive to the environment, then they essentially, the mussels, become just as sensitive. Right? If that host species is actually more resilient to environmental change than the mussel species, it's possible that the mussel as adults will die out because of those changes. And then it gets decoupled. Then it doesn't really matter. But it all depends, like their success, the mussel's success does rely on the fish host success. Resources being used to lure host.
You said horse.
Horse. No. Resources being used to lure host. Yes, the trade-off. Absolutely correct. That is another con associated with it. It's expensive to make a lure. And if you don't have to bring in a fish host in the first place, then you're saving resources and energy. And so there is an energy trade-off. Very, very good. For the Veliger larvae, don't need a fish host. That's right. Move with the current. Yes. Attach to any substrate. Yes. Reproduce quickly. Yes. Can spread further. Yes. These are all absolutely correct. More energy needed to care for the young with no fish host. They don't really care for their young. So I maybe I don't quite, I see a question mark on that one. So I'm not quite sure what was meant by that. Must feed on phytoplankton. Yes. Must feed. Yes. Might get eaten while free in the open water. Yes. That one is a really good one. The larvae drift downstream. Sure. Yeah. If there are more predators, it's kind of a pro-con type of a thing, right? Depending upon where they end up. It could be good, it could be bad. Unsheltered during development, yes. Dispersal is slower. Slower. I'm not so sure if you could say that it was slower. It happens in a different direction and it happens without any connection to other species. Larvae can't really control where they go. And nor can the larvae of the Glochidia, in that they are at the mercy of the fish, but I think I understand what you mean, that the larvae are dispersed broadly rather than being intimately protected by these fish hosts. So yes, we will, if higher risk.
Some stuff in.
Oh, you want to throw stuff in? Okay.
Yeah. So there's some discussion on the chat about this, asking for specific definitions, which we, as you may have seen, we tend to shy away from or don't volunteer because we're trying to get you to work on these things and define them by
Your own
doing
words.
them and using your own words, because your own words stick in your own brain much better. So basically the Glochidia, what we can do is I can tell you about the roots of each one of those words. The Glochidia, that's the form that is associated with the native unionid mussels. The lures, that jelly sac, or the snapping on, the push them into the gills where the little Pac-Man larvae latch on to gain that super guaranteed life of nice tasty blood and get locked in and depend, like you've been talking about. The Glochidia, the root of that is Greek, and it means arrow or barb. So if you think about glock, I don't know, you think about a projectile and you think about those projectile things arriving in the gill. The Veliger means sail. So that's the one associated with the zebra mussel that generally is going to get up and drift and go away. And one of the things that I liked thinking about when you were talking about the cons of pros of the Veliger, not depending on, and you've linked a lot of good thinking to dispersal, because it's a strategy built on being pretty much a generalist. So anywhere you land, you float is going to be good. So you don't depend on a species. But one of the things that as you know, with the story of the zebra mussel in the 20th century, is that because of that way of the Veligers, those sails floating up, is that they don't depend on us, but we have moved them. So they certainly interact with other species, and that has had dramatic consequences. So
Nice.
Arrows and sails.
Good job. Okay.
The slings and arrows of outrageous fortune, and all it be mussels.
Amazing. So we're going to clean this up for you and we'll post it for you, okay? So we'll remove ones. There weren't any that were like super incorrect, but what we'll do is we'll make them like uber correct and we'll post them. We'll post them for you.
This is the correctest answer.
Okay, cool. Thank you so much for that. Sorry about the breakout rooms. That's, but anyway, I feel
I
like...
hope nobody was in them when they disappeared.
Come back! Okay, cool. We'll follow up on the breakout room issue. So, given that, here is a question for you that I will throw on your screens right now. Take your time, okay? Read, relax, think. You want to play some music while we do that? Don't distract them. Oh, the other thing is...
You can whisper it in my ear.
I can whisper it in your ear.
Yeah.
Okay, I'll just put it on mute just in case. Oh no, but they can... Okay. I want to start using endemic instead of native. Feel like...
Sure.
Okay. I did. Oh, yeah.
It's an older word than how it's been used with people, but sure.
Yes. There we go. Oh yeah, we have to figure out the right answer.
Are the polls worth any marks?
No.
Marks in life.
Species that are dependent on very specific foods are less likely to go extinct. That's not true. Species with okay, we're looking for the true answer. More likely, species with Veliger larvae are more likely to go to extinct or higher levels of gene flow. Species that are dependent on very specific hosts are less likely to go extinct.
Some of you asking about the questions being cut off on the poll. Just drag out the window.
Yeah, make the window wider. And they're also here on the screen. Species with Glochidia larvae are more likely to speciate. Species with Veliger larvae are more likely to go extinct because adults cannot move to escape predators. Species with Veliger larvae are more likely to speciate.
Some other good suggestions in there too.
Okay, most of you, no, not yet. We'll give you your time. It's okay. Take your time.
We're at 70%.
Yeah. Do you have an answer?
To what question? This one?
This
No, I
one.
haven't read the question yet.
Okay.
It's true about either the risk of extinction or ease of speciation.
Super good.
We're looking for a true answer. I know what I think.
Yeah.
Said Dr. Smith to Dr. Jacobs.
Okay.
I know what
Good.
I think.
Yeah.
30 seconds?
Most of you have voted already. That's excellent. It's good.
Remember, just like your participation as a citizen of whatever geographic boundary you're in, vote early, vote often.
Wonderful. Okay, a few more seconds.
Certainly
There we
voted
go.
early.
Okay, so more than 90% of you have entered an answer, which is great. Remember, it's not so much about whether you get it right. This is setting an anchor for learning. So we're going to put a flag in it, whether you get it wrong or get it right. It's going to, just going through the exercise of choosing and working through it is going to be helpful.
Yeah, there was some chat in the chat
Chat in the chat.
about whether or not this was going to be marked. I said no. I think we said that out
Yeah.
loud. But it's good practice for aligning your thoughts, and it's also these are the kinds of questions that we write for midterms as well.
Yes.
This is our kind of question.
Okay, so I'm sharing the results with you right now, and you can see, and I'll put it on this screen here as well because it's part of the recording. So it's kind of a toss-up between C and E, which makes sense because it's one or the other, really. And the other ones, of course, if you voted for those, we're happy to put this, if you want, please feel free to put this question up on the discussion board where there's more opportunity to fully explain. But if you'd like, we can go through it, the two most common answers that were C and E, to explain them. If you're dying to know about another one, put a stamp on it, and we'll go through that one quickly as well. So I'm going to stop sharing the results.
I'm going to
close this up for a minute.
Should I clear the drawings, or are we going to use them to stamp?
Yeah, no, you can stamp on ones that, okay, D, I guess. Okay, so C and E were the two most common. So let's take a look at C. Species with Glochidia larvae are more likely to speciate because their populations are more easily subdivided. That is a true statement. So that was the correct answer to this question. The idea with speciation is that in order to happen, there has to be some kind of isolation. And we're going to talk a little bit about different types of isolation, whether it's spatial, so geographic, or temporal, over time, or resource use and those types of things. So, things can live in the same place, but still be separated somehow from reproducing, from gene flow, okay? So in this case, because the Glochidia are dependent upon their fish hosts, and because their fish hosts, can go upstream to certain places, their populations are more easily subdivided than the Veliger larvae mussels. Veliger larvae mussels, they move with the currents. They're just kind of this big blob that kind of gets bigger and bigger and goes along with the movement of the lakes and the rivers. And so they're not necessarily as sort of distinctly subdivided as the Glochidia larvae populations can be. Going to E, species with Veliger larvae are more likely to speciate due to extensive gene flow among populations. So gene flow is the exchange of genetic material across populations, and we'll show you some little simulations to help visualize that in just a minute. It means then that speciation is less likely with gene flow, because in order to have speciation, you have to have isolation. And gene flow is exactly the opposite. It's the mixing of things, right? So no isolation.
So speciation is you drawing your name in the sand at the edge of the Great Lake over the summer. That gene flow is the wave that comes in and washes it out.
Yeah. Okay. So somebody circled D. Species with Veliger larvae are more likely to go extinct because adults cannot move to escape predators. And this really focuses on the fact that all of the adults, both the Veliger larvae adults and the Glochidia larvae adults, can't escape predators. They're both sedentary. And so it wouldn't explain a difference between any of them, really. So I hope that makes sense. Okay, let's stop with the annotation, if you don't mind. For now, we can come back to it. And we'll show you some things. Related to definitions. Do you want to do this?
People are asking about what is gene flow.
This is, hold on to your hats. Here we go.
Here she comes. So these are some of the, we might as well populate it, the causes and sources of genetic variation. So as you're thinking about these, about genetic variation, either within a species, between species, the genetic variation, when we talk about that, we're referring to differences at the DNA level amongst individuals within a population of a particular species. So that's genetic variation. So that DNA shirt I was wearing the other day, it would have a G or a C or a G or an A, differing between different individuals within a population of Alex's wearing a t-shirt. So alleles, you've run into before, different versions of the same gene, and that one of the principles that we're thinking about is that genetic variation, one of the reasons that evolution can occur is because genetic variation can change within populations, within a species in various ways. Mutation, some of the big ones, mutation, natural selection, genetic drift, and gene flow. The primary source of that genetic variation is mutation. There's some other smaller ones, but mutation is a big driving one. Mutations, of course, are errors in DNA replication. So it's, as a biologist, I think we're predisposed, or we ought to be predisposed, to feeling okay about making errors, to getting incorrect answers on quizzes, because those errors are where the novelty comes from. It's where the learning comes from as a student, as a learner, and it's where novelty comes from in terms of being a species. But it's important to remember that these mutations do not occur in order to do something. They occur. They occur randomly.
Total mistake.
And it's not the giraffe evolving a mutation to grow a longer neck because the tree keeps growing taller than it is. That's a Lamarckian kind of way of thinking that we are not. So, is there more on this slide, or is that all?
Nope,
of this slide?
that's it. Yeah, well done. Okay. So
when we talk about mutations, there's, as being the principal source of variation, the driving source, random source of variation, the food for the fodder for this evolutionary mill. We have point mutations, which are what I was describing earlier, where there's a change in a nucleotide that can create new alleles, particularly if it occurs earlier on in the codon structure. There's chromosomal mutations, which are larger scale rearrangements or fusings of chromosome segments that alter gene order. There can be duplication events where bits of DNA, entire stretches of DNA are created, an additional copy is created. And then at a chromosomal level, again, there can be polyploidy or duplication events where a complete new set of chromosomes. Some of these are, as you look at the cause, kind of what drives these point mutations, can be chance error or errors in repair. Chromosomal mutations can be caused by radiation, ultraviolet radiation, or other factors. Unequal crossing over or during meiosis can produce a duplication. And polyploidy can come about because of hybridization. And hybridization, despite what you may have been talked about if you've been indoctrinated into the biological species concepts, hybridization is just ridiculously common.
yes. Yay. Cool.
So, natural selection, which you may have you've run into before, but this is, of course, the non-random difference in survival or reproduction amongst those individuals on the basis of those heritable characteristics. So it's by definition non-random. And we're going to talk more about natural selection in the coming days. And
Oh, sorry.
the another process of accumulating or stripping variation from a population can occur with genetic drift, where there's random survival, random differences in survival or reproduction amongst individual entities that's not on the basis of differences in those heritable characteristics. So one of them was about the randomness, but survival based on the characteristics, and one of them is random, but not based on the characteristics. Now, what can that mean? We have a drawing.
We have some drawings, okay? So this is where we kind of dive into really what evolution is. Evolution is that big umbrella term that talks about change, right? And it can happen due to random effects, as like related to the heritability or the allele frequency or the genes, or it can be selected. It can be non-random. And we're going to focus right now on the random things like genetic drift. Within genetic drift, though, there are some concepts that we'd like you to understand. So we've done some drawings, and hopefully they're going to work.
They will work.
They will work. Okay, let's give it a try.
We'll just see if the sharing of them works.
Yeah. It should be okay.
I can see what
You can see. Yay. Okay, so genetic drift. Remember, there has to be change with respect to the allelic frequencies, but it doesn't have to be because of those alleles, because they're more fit. So imagine, and this is going to bother Dr. Smith a little bit, we have a population of 6-legged spiders. Okay? It looked weird to have 8, so
Sure.
I just put 6. Anyway, so 6-legged spiders that are the same species. The only difference that we're going to be showing right now is body color. Okay, so some of them are red and some of them are blue. Now, of course, there's all sorts of variation that exists, right? The leg length, for example, body size, all of those things are just different. But we're going to just, for the purposes of showing you the difference, we're just going to focus on body color. So the population has 2 alleles for body color. One is red and one is blue. And genetic drift basically says that over time, for random reasons, not because blue is better than red or red is better than blue, but just because the frequency changes over time, okay? So it looks something like this, where, you know, whoever gets to breed into the next generation, randomly selected, they move on over time following the green arrow, and they're the ones that get to breed, and so they produce a population that has different frequency of those body colors. So there they are reproducing, and if we take a look then at the allelic frequencies afterwards, you can see that the ratio is different, but not because of fitness, not because the blue ones are better in any way, it's just because. So that's kind of like, I don't know, it was a little alarming to me when I learned about this. So what we'll do is we'll show you 2 extreme examples, right? Because, and in fact, this is an extreme example. It would happen a lot more slowly if it really just was genetic drift. But we'll show you 2 extreme examples where you can get, within a generation, a massive change. The first example is called a bottleneck effect, where something terrible happens usually. So it's usually a dramatic reduction in the population size, okay? And it isn't, again, because of fitness. It's just the shit happens hypothesis, right? Where something horrible happens, like a flood or like a tornado. And it just smokes like a whole bunch of the population, okay? And takes them out.
That's a biological term?
That's a biological term, okay? Not because the blue ones are better at surviving than the red ones. This was a tornado. Our spiders had no chance, no matter what color their body was, right? And then, taking a look at who survives, you can see that the population frequencies can change. And of course, this can be a lot more dramatic, depending upon randomly who was in the path of the tornado. So that's one way that you can have genetic drift. It's a mechanism of genetic drift. Okay, the next one is founder effects. So founder effects doesn't reduce the population in one place. but it actually creates a second population by a small group from the original population going away to basically colonize a new place or to start setting up in a whole new place where they never existed before. And therefore they split off, right? There is isolation between them. And it could look something like this, where on the mainland we have this distribution of 50-50. And just not because any of them are better at getting to the island, but just because a wind picks them up and blows them over, or, you know, they all stick to the warm feathers of a bird that brings them to the island. A group goes over to the island, and that changes the proportion of the alleles relative to the original population. So those are founder effects. Now, gene flow. This is different. And when you think about gene flow, think literally about genes flowing across populations, okay?
Not pantalon.
No, don't think about your genes. Okay, so think about genes flowing among populations. Here is the exchange of genetic material across populations, right? Where there is not genetic isolation. The founder effect broke these up. There was isolation. These over here were breeding amongst themselves. These over here were breeding amongst themselves. Gene flow basically is when all of a sudden, for some reason, they are swapping genes, okay? And they're producing offspring across the different populations. That's gonna look something like this. So imagine we have the mainland with 27%, 73% distribution. But because of a founder effect, because only red spiders made it across to the island in whatever scenario, we have 100% red. Now, again, they may be sitting on the feathers of birds, and birds may be flying across. They may get blown over by the wind or whatever. For whatever reason, we have individuals that are going from the island to the mainland, reproducing back and forth. And that would look something like this, right? So we have some blue ones that go, some red ones go back to the mainland. Everybody's kind of swapping genes. And over time, if they're going to be breeding amongst themselves, which is a condition necessary for gene flow, you can't just go and visit and then leave. You have to leave your genes. Over time, the allelic frequencies are going to be 50-50. And of course, there's going to be potentially some change going on in the original population. The idea with gene flow is that over time, it isn't necessarily that there's like more variation in a population. It's that the two populations over time become more similar in whatever variation they had, right? It kind of gets homogenized to the point where if there's enough gene flow, you're not talking about two populations anymore. You're talking about one. I hope that makes sense. But we are going to be posting these so that you can take a look and you can play around with it. More importantly, though, in terms of your learning, draw these yourself. Use whatever organism you're passionate about, and see if you can go from like one scenario to another to draw the difference between these different concepts. Hopefully the polls are going to work. Take your time because we just threw this stuff at you.
Can I throw something in before we go to this clicker question?
Yes.
So go back for a second?
Yes. Smith wants to
Some of you don't like spiders. That's weird, but fine. Some of you do like mussels. That's also weird, but fine. And I think one of the things about all of these examples as we're giving them, there's been some questions that we've received by email and on the chat as well about is the exam going to be all about these kinds of specific examples? Remember, the exams, the midterm exams, the lecture exams aren't going to be about the specifics. We're not going to invite you to define a Glochidia. That's not going to be it. We're going to talk about these processes. So each question will give you a scenario that will then ask you to think about and interpret for us that scenario in light of the concepts that we've talked about. So if you hate mussels, if you hate spiders, totally fine. You probably like something. You might like birds, which is like super weird.
Hey.
But translate all of these examples into pick your favorite bird, get a good image from Google image search your favorite bird, and draw these out. Recreate these slides with your picture of your favorite animal or plant.
Yes. Thank you. Very important. And slideshow, because now we can go back to the original deck. And we've launched the question for you.
And you're voting. And you're voting.
And you're voting. And you're voting. There we go. Gene Flow. There we go. Exam type question. Super. Well done.
Boop boop ka-choo, Dr. Robertson.
Hi,
Hello.
Dr. Robertson. Dr. Robertson is just joining us. Okay, so really good. We have about 75% of you that have answered. That's excellent. Okay, we'll just wait a few more seconds to get it up to at least 90%. Good, 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 good. Okay, excellent. We're going to end it there, and I'll share the results with you. So most of you said there has been C, so there has been a great deal of movement of mussels into this river from nearby lakes. Yes, that is the correct answer, in that it is not consistent with lower variation. The others are entirely consistent with lower variation. They would result in lower variation. We're running out of time for today, so if you do want a follow-up for why, please go to the discussion board and help each other. We will be posting this slide, of course, when we post our PDF, as well as it'll be available for you on the video. Just because D was the most popular incorrect answer, we'll tell you the species of mussel has remarkably efficient DNA repair enzymes. So we talked about mutation, and we said that those are errors or mistakes by random chance. And if you have DNA repair enzymes, then they would be going along and fixing those mutations, which means that it would reduce variation, or at least maintain the original variation, but it wouldn't increase. So that's why it isn't consistent with this particular question. Again, discussion board. We will be on it if you need any more support in working through this question. Thank you, thank you, thank you. Great. So homework. Stuff you can do as a result of this class. There we go. Lots of annotations in the background there. What stuff you can do as a result of this class is to work through the material again. And if you want, use this table to help understand some of the differences between all the terms. We threw a lot of vocabulary at you. You've had it, of course, online for a bit, but here's a table that if you fill out will hopefully help you identify the big differences and the consequences of each of these terms. So this will be available to you to use as a kind of study guide to bring you through some of your learning.
It can be a matrix that you take your preferred lovely animal and that you expose them to. So you've got your...
Elephants.
Oh, sure. Okay, elephants. And then you expose them, create and draw out a situation of genetic drift. Talk about what would happen, draw it. Is the genetic variation going to... or here, or here, or here?
Yeah. Okay, and then if you want, and for me this was like so useful, but it's completely up to you. Here are a whole bunch of the words that we threw at you today, specifically. And if you want, you can take these and you can create like a concept map, flow chart of like start at the top with evolution, right? Evolution is the change that we see in the world, right? Then see if you can take these things and kind of go from there, match them together, and see if you can kind of find the next big concept and then the ones that are nested within those. So if you'd like to play around with this, I've seen some awesome concept maps that have come from these words and these terms, and hopefully again that will help you understand how they're all related. We can talk a little bit about that next class. Couple of announcements. Project Tree. We're going to be starting next week with the first images. So if you would like to participate, please make sure that you take the quiz. It's not really a quiz. It's just basically a survey to make sure that you've read some of the safety considerations, like don't go out at night by yourself, those types of things. And then once you take the quiz, it will give you the instructions for how to participate in Project Tree. And if you're not going to participate, it would be so helpful if you could go into the quiz and just say that you're not going to participate. We have to submit a list of students who are participating to the university for insurance purposes, blah, blah, blah. So it would be great if you could do that. For those of you who are enrolled in SAS, if you would like any accommodations related to your registration at SAS to be put in to the assignments and the quizzes that we're doing, please do get in touch with Dr. Robertson, and we'd be more than happy to make sure that you get what you need. I think that's it. Yeah. Wonderful.
Be safe.
Be safe.
Be physically distant.
And take care.
And socially connected.
And we'll see you next class. Have a wonderful weekend. And we're just going to shut down for a minute, take a breath, and then we'll be back up for student hours.
Ta.